started. Just a couple of housekeeping thing. One is please make sure your name is showing up in Zoom. Looks like I think everybody's already done that. I think you're automatically muted, but if not, make sure you're muted during the presentations. If you want to submit questions throughout, you can do that in the chat, and then we will also have live question and answers. And for that, we ask is people just click on the raise hand on the bottom, and then we will call on people. Hopefully we'll get to everybody. This session will be recorded. And Maggie, any other housekeeping things I need to mention?
No, I think that covers all.
Okay. And we'll right up front thank both Maggie Gopal and Anthony Jimenez for all their work in planning this and setting it up. And to Nora Wong, of course, for helping organize, as she always does. All right, so we have a interesting scientific symposium today. We are predominantly talking about genetics, and we have a few speakers, two about the genetics, then we have one patient and family representative. Then we have a short presentation of a new study related to the vagus nerve stimulation. And that'll be it for today. So we're going to start with Danielle deCampo. So Danielle is an MD PhD from University of Rochester, where she studied neurobiology and anatomy. She also has an MS in biomedical and chemical engineering. She did her pediatrics and child neurology training at Johns Hopkins in Baltimore, then did a PEDS epilepsy fellowship at the Children's Hospital of Philadelphia, better known as CHOP. She is still at CHOP, where she's now an attending epileptologist and assistant professor of neurology. And her primary focus is understanding the influence of genetics on epilepsy, Norse, and fires. So Danielle, I will let you take it away and share your screen.
All right, thanks so much, Larry. I'm sharing my screen. All right. Can everybody see my screen okay?
Yes, perfect.
All right, well, I want to thank you guys so much for giving us the opportunity to speak with you a little bit today about the genetic underpinnings of Norse and fires using whole genome sequencing. This is some preliminary data, so plenty of things we still have left to do, but we're really excited to talk with you a little bit about what we found and kind of the implications for our next steps. So I'd like to divide this talk kind of into three sections. The first is really trying to have a better understanding of what it means to have a genetic diagnosis of epilepsy, a little bit about how we go about coming up with this diagnosis, and an interpretation of one class of variants, where variance means a gene with a certain sequence change. And the specific ones that I'd like to talk about are variants of unknown significance. And this is really relevant, I think, in understanding fires in Norse, and that many of our variants that we've identified in the past have been VUSs. The second part of my talk will really focus on looking at what we know about genetic findings in fires in Norse in terms of prior genetic studies, and also kind of the theme that we've been encountering in our last couple studies, which has been that compared to other forms of epilepsy, the genetic yield in Norse and fires has been historically lower. And then I'd like to finish with going through a little bit of our preliminary data that we've gathered, looking at whole genome sequencing in individuals with fires in Norse. And this is comprised of 65 individuals that have graciously been shared with us through the Norse Institute, including the multi-site center study, as well as the biorepository. So starting off in that first kind of category about how we identify genetic forms of epilepsy. In general, over time, we found an increased rate of gene discovery in epilepsy, and this has really changed over the last couple decades. with next-gen sequencing. And right now in our current epilepsy clinic, our yield of finding a genetic diagnosis for individuals with developmental and epileptic encephalopathies is as high as 30%. So when we have a patient come to our clinic to determine whether or not their epilepsy has a genetic basis, we kind of divide it into two steps. The first is really identifying what type of test we want to order. And that really comes to looking at their clinical history, studies, and physical exam findings, and then determining if we want to do a more narrow focus type of test, like a gene panel, which has a set of genes associated with epilepsy, or something more broad, such as whole exome sequencing, which is a little bit more comprehensive in terms of other findings. Once we send off the test, we get back the test and from the labs, and we can get some different findings that have been classified according to the ACMG, which is the American College of Medical Genetics, that have designated 3 different kinds of classifications. One is pathogenic or likely pathogenic. So these are kind of positive findings that indicate that what we find is likely to explain our disease of interest. Negative, which is benign or likely benign. And then this third category, which is variants of unknown significance. And this is really where our homework in our lab and also in clinic really takes place, because variants of unknown significance is really an interesting category in that the majority of these, just by definition, have insufficient information to designate really whether or not they're disease-causing or explanatory of the process or not. These are a common finding, and we expect to find these in our gene panels as well as whole exome sequencing. And one of our jobs as geneticists and genetic counselors is to really take a deeper dive into looking into these VUSs to see if we can determine whether or not they're suspicious or not. And there's a number of criteria that go about helping us to interpret VUSs. And this is kind of like a list of some of these things. So one is determining what type of variant it is, whether it's a missense, meaning a change in a single base pair, or whether it's a protein truncating variant, where you can have a stop codon that results in an early termination of the variant sequence and absence of protein. We also like to see whether or not this is a variant that has a high frequency in population databases of unaffected individuals. We look at computational models about how that change in the base pairs or sequence can potentially lead to a deleterious change. We look at databases to see if it's associated with a clinical disease, this particular gene spelling. And then a nice luxury that we frequently have in our clinic dealing with kids is the ability to do segregation studies. So looking to see whether a variant is present in an uninfected parent or not. This can give us a little bit more information, because if we see this variant in an unaffected parent, it gives us a little bit more confidence to say that it's less likely to be suspicious. And I think understanding VUSs is really important and relevant for us when we are starting to look at the research and also the findings that we get in FIRES and Norse. And that is because when we think about VUSs, we have to really think about how we interpret them. And when it comes down to the VUSs, the majority of VUSs that we find are really not suspicious. And so this is kind of a figure showing the gradations of VUSs in terms of how likely they are to be a suspicious kind of gene or explanatory of the disease. Over time, as we learn more and more information about a particular variant, it's possible to reclassify the variant as benign or as pathogenic. And what we found is that typically the majority of VUSs are deemed ultimately to be benign or likely benign and not to be pathogenic. So maybe about 10% you'd say could be reclassified as pathogenic. And I think it's important to keep this in mind, because I think when we look at studies talking about the genetic findings in FIRES and Norse, I think it's important to keep in mind that, it's important to look at it from the research perspective, but also to not attribute too much in terms of saying definitively that it is causative of a disease. We've definitely had cases of children, for example, one who had SCN1A, that came to us as a diagnosis. And then on further discovery, we decided that this variant didn't really seem to be explanatory of the kid's condition, and his epilepsy was more likely to be related to a malformation. He underwent resective surgery, And it really, I think, illustrates the fact that sometimes the VUSs can sometimes delay the proper care of kids when we're not carefully thinking about how likely they are to be related to disease. And so as I've kind of tried to allude to, I think that this is really relevant to Norse and fires in terms of the prior studies that have been identified. This is a list of all the previous gene findings that have been identified in individuals with Norse and fires. These are largely case reports or case series. And what you can see here is that you may recognize some of these genes to be genes that are involved in epilepsy. And I think one thing that is a little harder to understand is that sometimes it's hard to follow the particular variants in the literature, and sometimes we have insufficient information about these findings. For example, in some of these case reports, the precise variant or the coordinates of where this gene is, this sequence change in a gene is located isn't well described. And then its classification in terms of ACMG is also not always provided. So whether it's a pathogenic change or a VUS. And then lastly, a lot of these findings are VUSs. And again, I think describing our VUSs and reporting them is very important, especially in the realm of research where we're looking for new changes. But I think it's also important to be able to clearly state that these are VUSs, and so our interpretation is somewhat limited. When we take a look at larger genetic studies, typically our finding has been that compared to other genetic epilepsies, our yield of genetic discovery in Norse and fires has been historically low. So in one study done by our lab, we looked at 50 children with fires who had whole exome sequencing that was done. We had the luxury of having some patient-parent trios, meaning that we were able to do further interpretation of some of the variants using parental information. And through this study, we were unable to identify any pathogenic variants in epilepsy-related genes. We also looked at HLA sequencing and did not find any prominent alleles within this study. And this is, again, to just kind of drive that point home. This is a figure from a meta-analysis that's looked at a number of different studies that have identified genetic forms of epilepsy. And you can see here that the discovery rate in each of these studies really seems to vary, but in general, overall, the discovery rate is about 24%. And this is our study from 2020 that looked at the children with fires. And you can see here that we have really kind of a lower yield. And I just think that this probably means that these children or individuals with Norse or fires may really represent kind of a unique subpopulation that we may need to think about a little differently than we have with other types of genetic epilepsies. More recently, we've done another study where we have looked at children here at CHOP who were diagnosed with fires. These children were identified using a lot of electronic mining of data of our electronic medical records. And Ingo is going to talk with you guys a little bit more in the next talk about how we do that. But we were able to find about 25 children over about a 12-year span and found that about 23, so 92% of these children had genetic testing. When we looked through that testing, we were unable to find any individuals who had pathogenic gene findings for fires. This is a figure to kind of show the different types of genetic testing that each of these individuals underwent. And you can see here that it really isn't from a lack of genetic testing. So for many of these children, they underwent lots of different forms of genetic testing. So I think the finding that we didn't find any pathogenic variants is pretty striking. We then wanted to look at kind of a broader sample looking for more Norse-related population. And to do that, we took data from children that we see in our epilepsy genetics clinic and again used the electronic medical record strategies to identify those who've been diagnosed with status or refractory status epilepticus to further define if we could identify any individuals with Norse. Overall, our cohort at that point had 36% that had a genetic diagnosis of epilepsy. And when we looked in more detail, we were able to identify 6 individuals that did have a genetic diagnosis and that did meet criteria for Norse. And these are in some very well-known epilepsy genes, including CACNA1A, PCDH19, RANBP2, which was actually twins, and KCNA2. So again, we're looking at kind of genetic epilepsies in general, where the yield is high, and then for Norse and Fires, where that yield is lower. But I think obviously we're still trying to grapple with a lot of different genes, a lot of different processes, and it's important that I think we continue to look for these because we are finding some good findings that implicate some genetic involvement in Norse and Fires. So that leads us to kind of our current study that we're working on right now, looking at whole genome sequencing in individuals with Fires and Norse. This is a group of 65 individuals who had whole genome sequencing done. And with these 65 individuals, we did an analysis to look for gene discovery, basically looking for different types of variant subgroups. So to just briefly introduce whole genome sequencing in general, when it comes to our clinical approach to genetic testing, whole exome sequencing really represents our gold standard for comprehensive genetic testing. In whole exome sequencing, we are looking at coding sequences of human genomes without analysis of the non-coding regions, and this covers about 50 million base pairs. When we do this, we analyze really a small fraction of the genome, so 1 to 2%, but still are able to cover 85% of disease-causing variants. Typically, our yield for this is 25 to 45%. Now, when we start talking about whole genome sequencing, this is a relatively newer approach that's primarily performed on a research basis, although it's becoming much more clinically available over time. In terms of whole genome sequencing, this really looks at the whole genome. So that's 3,000 million base pairs that we're looking at. And of course, with all this extra data, we also generate a lot more variants and a lot more variants of unknown significance that we have to view. The good news about whole genome sequencing is it also gives us the ability to look at the non-coding regions of the genome and also assess for any sort of discovery in those regions known as introns. So in this study, we wanted to look to see if we could identify any genetic etiologies in Fires or Norse using whole genome sequencing. And to do this, we used a population gathered from a multi-site study of Norse and Fires, as well as the biorepository that is established through Yale. So this is really kind of our ability to take a deep look at all the data that's been collected through generous donations from a number of different people who really are trying to help us better understand this. And I think we're all very grateful to have had the opportunity to work on this sample. So in terms of the samples that we have acquired, we had 72 individuals that were identified with Norse or Fires. 7 samples that were excluded. And then that left us with 65 individuals who had whole genome sequencing done. 52 were enrolled in the Norse multi-site study, and 13 came from the Norse Fires biorepository. In terms of a little bit of the demographics of this population, the majority were adults, and there was a combination of individuals with Norse as well as Fires. You can see here that for Norse, the median age of onset is a little bit higher than in Fires, which is, I think, expected. And when we look at etiologies, also as expected, the majority of individuals who had Norse or Fires were ultimately diagnosed with a cryptogenic form. And this really seems to be kind of what we're working on is to better understand these. But the other etiologies that were described are listed here. And you can see that in addition to a couple of the other ones, that there are at least 2 genetic conditions listed, and that's RANBP2 and POLG. And I will go through that in a little bit more detail going forward. This is a slide that really shows the workflow of how we go about analyzing whole genome sequencing. That really comprises 2 pieces. So one is kind of the upstream analysis that involves a lot of manipulation of the raw data and presenting it in a way that provides us with a number of variants to review. And that is really the downstream analysis. And in our lab, we have a lot of very experienced people that we've been working with. Shiva and Ian have been 2 of the data scientists working with me, as well as Katie Rose Sullivan, who's our genetic counselor, who has done an amazing job in helping me digest this very, very large data set. And to just really illustrate how large this data set really is, I will tell you that when we finished and got to the end of this workflow, we ended up with 11 million variants that needed to be reviewed. So obviously, this is a large number of variants that we really have to do a little bit of digestion and filtering to get to a more manageable number. And to do that, we do kind of a standardized process that, you know, people do in all sorts of clinical and research settings. One of it is trying to see if we can weed out some of the ones that are more common or in unaffected individuals that are less likely to be suspicious. We do have a number of tools and databases that we use, and I'm just going to briefly introduce a few of them that may come up in this talk. So NOMAD is a database that has unaffected individuals. We use this to basically look to see what the frequency of our variants are in this population. Gene constraint scores, which measures how intolerant a gene is to change. ClinVar, which is an NIH database where labs or clinical kind of laboratories can submit interpretations about how likely something is to be benign or pathogenic. A CAD score is a score used to predict how deleterious a gene change may be. And then OMIM, which is also a database that has linked genes with associated phenotypes and given reference numbers. So we use all of these in conjunction to kind of help work through some of these variants. And then we also use some general filtration criteria. These are pretty standard criteria, but basically we want to make sure that we are looking for variants that are not common in unaffected individuals, as well as making sure that we filter out any sort of variants that may be due to artifact. I'm just going to mention briefly two caveats of this whole genome sequencing study that we've done. So again, we're starting with 11 million variants. And I think one thing that's important to keep in mind is that many of these are going to be VUSs. And as we've talked about before, the majority of VUSs are not suspicious. And I think even when we do have VUSs, you know, it's important to realize that we cannot definitively say that something is explanatory of Norse fires, although we may have a higher suspicion. And then the second piece that I think is a little bit challenging when we're doing whole genome sequencing, especially on singletons, is that interpretation of autosomal recessive conditions is limited. And that is because in autosomal recessive conditions, you need to have one gene change on each copy, one from dad and one from mom. And based on our variants, it's a little hard to know when we have two changes if they lie on the same copy, so on mom's copy or on dad's copy, or if there's two changes on two different copies. And so we refer to that as cis and trans. So I think that becomes a little limited in our interpretation. And so sometimes it's a little hard for us also to know, even if it is a pathogenic finding, if the individual is just a carrier or if there's really a disease that's underlying it. But with those caveats, I think we have a lot of data that we get to go through and look at. And so that brings us to kind of how we went about analyzing whole genome sequencing for these 65 individuals. So to do that, we went about looking at variants using three criteria. And so you can think about these as kind of three different variant subgroups. So in the first step, what we wanted to do was just kind of look at what are any sort of pathogenic or likely pathogenic variants that we find at all in individuals who have Norse fires. So these are classified by ACMG as pathogenic or likely pathogenic disease-causing variants. In the second criteria, we are going to look at protein truncating variants. And the reason we have decided to look at these variants is because these are really prime candidates for disease. Because in this kind of variant, what you have is an absence of a protein caused by an early termination of a sequence. And so I think these are definitely a good promising candidate area to study in terms of finding new genes. And then lastly, we wanted to look at genes involved in epilepsy. Obviously, this makes sense. And to do that, we use kind of our own little virtual gene panel that we've used previously before, including the number of majority of well-known and well-described genes that are involved in epilepsy. And then lastly, we wanted to make sure that we covered any variants that had been previously described in Norse fires. So starting with our first criteria, these are pathogenic or likely pathogenic variants. We had to further filter things. We decided to keep intronic variants, filtered out nomad variants, and then looked really for autosomal dominant associated variants. And even after doing all this filtration, we still ended up with 152 genes to review with 213 variants. So this is a short list of the genes that I'd like to kind of highlight from this criteria analysis. So here you can see two variants in two genes, TUB8 and TMEM127. Neither of them were very familiar to me, but I will go through them a little bit. And then the third finding we had was one in POLG, which I think is really interesting, and I think something that just really requires a little bit more in-depth discussion. So I'll save that towards the end, as it also became relevant in our criteria 3. So just to briefly go through these two gene variants. So TUB8 is a missense variant, and the reason that we thought it was interesting was that it was found in 2 individuals. This is a gene that's involved in microtubule formation. When we looked a little bit further, it seems that this is a gene that is really only expressed in oocytes and early embryos and is implicated in mitotic arrest, but not really expressed in the brain. It has been identified in women with infertility. TMEM127 is a protein truncating variant that was considered likely pathogenic. This is a gene that's involved in cell growth and survival, and has been identified in individuals with pheochromocytoma, which is endocrinologic tumor that develops from the adrenal glands. And I think what's important to illustrate from both of these genes is that, omics data really provides with us a lot of variance. And sometimes it's helpful, and sometimes it's a little hard to interpret how that helps us or if it's relevant at all. But I think it's important to mention that these are kinds of the things that we go through in terms of looking at all these variants. The second criteria that we did was looking at identifying protein truncating variants. So with these, we are identifying frame shift, nonsense stop, and splice site variant mutations. We also did a number of filtering methods for this to make sure that we were able to work with a manageable variant list. And in this, we found 95 genes with 110 variants. And these are the two genes that I wanted to describe that we kind of ran into. One is KMT2C, and the other is TOP2B. So KMT2C was an interesting gene. This is a gene that's involved in gene transcription, modifying chromatin structure. And clinically, it's associated with a syndrome called Kleefstra syndrome. This is a disorder that we see in childhood. It's rare. It's associated with hypotonia, intellectual disability, and specific facial features, but also is involved in seizures as well as structural brain abnormalities. It can also have system involvement. And so this kind of leads us with the question of how is that related to Norse fires? And I think really the answer is we don't know yet. So we do know that it's expressed in the brain. We do know that it has some implication in seizures, but in terms of how that's related to Norse fires, I think that's still a big question, if it is at all. So I think these are kinds of the things that we're coming across, but things that I think should report and keep in mind when we're looking at things in the future. And then the other gene that we looked into was TOP2B. This is also an interesting gene in that it encodes for type 2 topoisomerase and is important in DNA replication and transcription. This is also involved in another syndrome that's called BILU syndrome. This is a B-cell restricted immunodeficiency syndrome, and I thought that was a little bit interesting given the implications of the immune system and inflammatory processes in Norse fires. In terms of seizures, I think that this has less to do with seizures, but it is something that does seem to involve the immune system and something that we did find this kind of suspicious variant in our data analysis. And then third is I'd like to identify and look at variants in the epilepsy gene. So these are coming from a kind of a virtual gene panel curated list. And variants in this are ones that we can look into a little more detail. And then the second part is making sure that we cover the genes that have been identified in Norse and fires. I will say that the majority, I think all of them actually were in our epilepsy gene panel, but I would just like to emphasize that kind of we made sure that we went back through the literature to identify and cover for those genes in our whole genome sequencing as well, to really kind of continue to build on prior data that's been collected by others. And so when we did this, we also did some filtration, leaving us with 80 genes and 202 variants. This is a table showing you the common kind of top 4 or so findings that we found using this approach. I put in the red box 2 genes that have been previously identified in individuals with Norse and fires. And I think it's important to keep this in mind as we go forward when we're trying to really look at these variants. So in terms of these 4 genes, the first gene is one that you may all know of. This is UBE3A. This is a gene that is associated with Angelman syndrome, as many of you know. This is a gene associated with neurodevelopmental delay, ataxia, epilepsy, and speech difficulties. When we looked back at the patient to see if this was clinically matching, we found that this was a 27-year-old who was diagnosed with fires without any significant past medical history. So I don't think in any way a slam dunk for Angelman syndrome, but it was there. It was a finding that we had. So I think it's important, again, to just kind of keep an open mind in terms of other potential ways this gene may be working. Another gene that we found was GRIN2D. This was a missense variant. This is a gene that encodes for glutamate receptor, NMDA type, and is associated with developmental epileptic encephalopathy. We do see quite a bit of these kids here in our clinic. This was identified in an individual that was 83 years old with Norse and no past medical history. So again, kind of the same idea as our prior individual with Angelman, that I think it's something to think about, but doesn't kind of give us the whole spectrum of what we were expecting to find. We also found a variant in CACNA1A. I'd have to say CACNA1A is one of the variants that seems to keep popping up over and over again, in terms of how pathogenic each of the variants that previously has been reported is a little variable, but it is one that is popping up. And this is a gene that's important in voltage-gated calcium channels. It's associated with several different disorders, including episodic ataxia, familial hemiplegic migraine, and developmental epileptic encephalopathy. When we looked back at our samples to see what the clinical history was for this patient with this variant, we found that it was a 5-year-old with fires with no past medical history, although it did present with some eye pain and headache a few weeks before onset. And I think it's a little hard to know exactly the outcome of this kid in terms of the diagnosis of CACNA1A, but definitely something that's interesting, and I think something that increases our suspicion. Relin was another gene variant that we identified. This is a gene that's important in signaling and post-mitotic migrating neurons early in development. It's most classically associated with lissencephaly, which is a brain malformation disorder with severe seizures and infantile spasms, but more recently has been kind of broadened to include neurodevelopmental disorders and autism. When we looked back to identify the clinical findings of the individual with this variant, we found that it was a 26-year-old female with Norse who had a history of GTC 2 years prior to her onset of Norse in the setting of intense pain. And then the last 2 genes that I'd like to review, I think I'd like to spend a little bit more time on because I think they are pretty interesting, and I think they do have a lot of potential in terms of further studies, Norse and fires. And the first one of these genes is POLG. And you may have noticed that POLG keeps coming up. It's come up in the past. And I think we need to spend some time really thinking about this gene. This is a gene that encodes mitochondrial DNA polymerase. And in doing so, it's involved in helping with replication of the mitochondrial genome. While we typically think about it with Lee syndrome, it's important to keep in mind that POLG is associated with a wide host of different disorders that span across multiple different ages. And so when we think about what Lee syndrome is an autosomal recessive condition characterized by epilepsy, liver failure, and developmental delay, typically presenting around 1 year of age, although juvenile onset cases are reported. And one characteristic that I think is relevant for us is that this disorder can have a really explosive seizure onset. This is a figure from a review that kind of illustrates the different disorders that can be associated with POLG. POLG can either come in autosomal dominant form or autosomal recessive form, and depending on which of these inheritance patterns can present with different phenotypes. So other things that could be suggestive of POLG would include things with difficulties with eye movements, neuropathy, so sensory changes, and relevant for this discussion is neurogastrointestinal dysmotility. So I think just illustrating that there's a lot of different presentations that this gene can have. This is a table that's showing all of the different variants that we found in POLG through our whole genome sequencing. And you can see here that it really is a lot. And so each one of these lines is a different variant that was identified in different individuals. And for these first three, I think these were probably the most highly suspicious for individuals who had actually POLG, especially the first one, which I think is pretty straightforward in terms of that diagnosis. This is an individual who was homozygous, meaning had one of these copies on each of the chromosomes. We found it listed twice. This is a recurrent variant that occurs in POLG and is very well established with POLG-related disorder. This was an individual who in our sample was an 18-year-old with Fires, but presented prior to this with paresthesia and blurred vision, and also had a history of some learning difficulties. So I think that this sounds very consistent with a presentation of POLG based on the information that we have. When we look at this sample, I think this one is also very suspicious. This individual had 3 variants, 2 that were pathogenic and 1 that was uncertain, again, because we don't have segregation studies. It's a little hard to know whether all of these variants lied on 1 copy or on 2, but I think we have some pretty good information. There was an individual who had POLG. This was an 18-year-old with Norse and no significant past medical history. So I think it is possible. And then last, this one was also an individual who had 2 uncertain variants and presented at 31 years of age with Norse, but did have a history of being on TPN due to gut motility since the age of 2. So I think in general, in terms of how suspicious we are for POLG, I think this one is pretty straightforward, and the other 2 I think are highly suspicious, although I think it's hard to definitively know, but I think there's a good chance that these are important for us to think about. And then these were other individuals. You can see here that some individuals only had 1 pathogenic change, so that would be considered carrier status, meaning that they wouldn't go on to have the disease, but could pass it on. And then some still remained uncertain, meaning we don't have enough information either way to say whether or not it's pathogenic. So the last gene that I'd like to wrap things up with that I think also merits some attention is RANBP2. There was a patient who was identified as having RANBP2 in this sample. RANBP2 is associated with acute necrotizing encephalitis. And the variant that we identified in RANBP2 in this case was a variant of unknown significance, or VUS. So just quickly to review, ANEC, or acute necrotizing encephalopathy of childhood, is an encephalopathy, catastrophic kind of disease that can follow after febrile illness. Like POLG, it can kind of have an explosive onset for seizures. It can leave you with focal deficits, and it has some very well-described features on the MRI, including bilateral signal intensity changes in the thalamus and the brainstem, and sometimes in the basal ganglia. And it is autosomal dominant. That being said, what's interesting about this gene is that it has incomplete penetrance, meaning that even if you have this copy that is pathogenic, it does not always necessarily mean you would go on to have ANAC. So it makes it a bit more challenging to really understand this disease and this gene in general in terms of Norse and fires. Typically, it is sporadic, but in some cases, it can be associated with RANBP2, and then it's considered more familial. So when we looked at this VUS, we identified that this was an individual who had fires at 16 years of age, also had a history of Hashimoto's thyroiditis, and was diagnosed with ANAC. We were able to look at the MRI findings that were reported, and I think there is quite a bit of information here to suggest that this is highly suspicious for ANAC. And while this is a VUS, I think this puts us in a little bit of higher suspicion that it is related and relevant and explanatory. But again, this becomes the problem with VUSs is we just can't say definitively, but we can have higher suspicions. And so that's typically how we present the information to patients, but also just to think about it when we're doing our research studies. So that's kind of the overview of our preliminary data that we've been working on looking at these whole genomes. Just to summarize, these are the genes that we identified using the 3 criteria. There's some genes that I think are a little harder to make sense of than others. And there's some genes that have come up again, and some genes that I think really do have a very strong potential to be important in fires of Norse, including POLG, RANBP2, and CACNA1A. And so to leave you guys with a couple take-home points, I think it's important to keep in mind that when we do omics data, we really do go through numerous variants that we need to review, and there has to be kind of a standardized process in which we do that. VUSs represent the majority of findings in Norse and fires, and because of that, I think it's important that we maintain some level of suspicion when we're looking at findings. And I think it's also important that the best we can to give as much information about a variant as we can when we are reporting information about a patient in the literature. There is an emerging storyline for some epilepsy genes, mostly RANBP2 and POLG. And then in terms of next steps, I think the thing that we have been most focusing on at this point and in all prior studies has really been looking at epilepsy-related genes, where there's been really only a handful of studies that have kind of approached looking at other classes or groups of genes. And I think one area that's ripe for investigation that I think we've all talked about but haven't started working on yet is looking at genes involved in immune or inflammatory conditions or processes. And I think that this is definitely something that we need to explore next and is kind of part of our goals for the future. So with that, I'd like to just give a few thanks. This project really wouldn't have been possible with some members of my lab. Katie Rose Sullivan, who I work with closely in the clinical and research side. She's our genetic counselor. She is great. She has taught me so many things over the past couple of years, and I appreciate working with her on both aspects in research and in clinic. Shiva and Ian are also very instrumental to making sure that we can do the data analysis and do it in a really rigorous scientific way. Both have been very instrumental in working in both the upstream and downstream processing and have been readily available whenever I've had a free minute out of clinic, and I'm very thankful for that. And then also to my mentor, Ingo, who has been great to work with over the past couple of years. And then also for Dr. Hannon, Dr. Hirsch, and Dr. Gaspard, who have been gracious enough to help us share this information and to help us get it out to you guys so that we can continue to look for other answers for Norse and fires. And with that, I think I'm ready for some questions.
Fantastic. Thank you, Danielle. That was great stuff. A lot of information too. Maybe Ingo, I'll let you, maybe you can just summarize what's been going on in the chat. We have 4 or 5 minutes for questions, so not a lot of time.
Danielle, I'm going to actually just unmute my camera, open my camera and use your microphone. Okay. So I think Anna and I had a bit of going back and forth about disease mechanisms and how to choose disease. And I think this has been a theme, especially in Norse and fire genetics, ever since we kind of started this. Is there any smarter way, or is there any different way how we can find genes that we can prioritize or can attribute? The analysis that Danielle has done is typically like the first analysis where we actually not so much think about like biological mechanisms, but think like prior evidence for genes. And this is important because when you zoom out and look at other disease aspects, this is how typically these genes are found. And then yeah, I would completely agree with you that there's basically this adds another twist to the Fires Norse story. I think there's some interesting findings there, right? And this is to be continued. And I was talking about a question popped in from. I should use my own microphone. That's it. Would you mind unmuting yours?
Yeah.
And we just need to unmute the sound off. All right. Is this better now?
Yes.
Yes,
Much
yes.
better.
Apologies. We're trying to do a good thing and then it turned out bad. So there's a question with for the POLG variants. Danielle, and this is we had one patient who had one individual who had a single pathogenic POLG variant with no other variant. And Marissa asked, what do we think is going on there? Is this incomplete penetrance, or is this some kind of an infection that may have triggered some metabolic stress?
I think in terms of understanding what this one individual with one pathogenic copy has, I think it's a little unknown. Clinically, if we see a patient who comes to us because of a history of epilepsy, and we find one pathogenic copy, at this point, clinically, we would say that that person is a carrier, but that is not likely to manifest kind of any POLG-related syndrome. So at this point, we would tell them, if you were to think about having a family in the future, there would be a risk for potentially passing this on. And if you were with somebody who also had a particularly pathogenic variant, there'd be a chance for your offspring to have POLG. But in terms of you as an individual, this is less likely to be an issue. We don't feel that this is clinically relevant. Now, when we're in the research realm, I think things are a little bit different to make sense of a pathogenic variant. Although I think, I just don't think we have enough information to say whether one pathogenic copy could cause something. I don't think we've really been able to show in other areas of POLG that that's the case for epilepsy.
And then one question came in, and probably this is more for Larry from Sophie, about basically whether samples that have been donated to Yale have been used for testing.
Yes. So these 65 samples probably does include that. We can track that down for any given individual. There was also a question about whether we're still taking samples, and the answer is absolutely yes. Both in the multi-center study, if it's an active patient at a center, it's in our multi-center study, but we can also take samples from all over the world as part of the biobank now. And the best way is just to contact us on the emails, which is norse@yale.edu. And we're taking not only acute cases, but also prior cases and some controls.
And then
Go ahead.
the random two questions keep popping in, Danielle. So this is a question. RANBP2 is very interesting, especially that the presentation of encephalitis can be a viral trigger. Do we know if there's been any comparisons of pathological findings in this and Norse fires where changes were found? So I think the question we need to rephrase is, what is the overall yield and what is the overall kind of frequency of RANBP2 variants in Norse fires?
So within the sample, we found one VUS, so not clearly pathogenic, but when we looked through some of the clinical data, including the MRI, I think, it seems consistent with a finding of ANEC. So I think there's a good possibility that we could attribute it to RANBP2. But, it is a VUS. So I think that's just something that you have to explain clinically and also in the research world. Within our own data sets, we have identified a set of twins who presented with RANBP2, and both actually had ANEC. So I think that that's really the majority of findings that we have. So I think it may not be a high percentage of individuals with Norse fires, but I think it is definitely something when we're thinking about genetic etiologies that may be important and relevant. And I think given the fact that, there are kind of this like possible encephalitis component to it, it does speak also to kind of the immune inflammatory processes that may be underlying Norse and fires.
And Lena just rephrased the question. The question was about comparisons in brain pathology, if available. I can maybe just quickly answer this. We don't have the information. I think the information. I think ANAC is first of all, very typical imaging finding. That would hardly be missed in anyone who has Norse fires, and there is no strong evidence that this is found either on the pathological basis or imaging finding in the majority of Norse fires cases. And then there's basically a question from Paul that I'm going to put back to Larry about how many people present with Norse annually?
Yeah. Some people are working on trying to get more accurate numbers on that, but the short answer is a few thousand. But we think it's underdiagnosed. All right. Well, again, thank you, Danielle, and we're gonna move on. I'll make one other point. There's a question about individual results. So these studies, when something is sent to the biorepository, we do give some acute cytokine results back to the referring center, to the physicians. But other than that, it's all just part of research, and we don't give individual results. If an actionable genetic finding is found, then the referring physicians would be notified. All right. So our next item of business is Paul Brady, who has been kind enough to join us to give us the all-important patient and family perspective. And Paul's going to speak about life with his wife, who is a Norse survivor. So, Paul, take it away.
Thanks, Doctor, and thanks for answering the question. I think my wife actually dropped that in the chat herself, watching in the other room, so appreciate that. Thank you all very much for the opportunity to be here and provide a little anecdotal information. My wife and I live in New York City, and she was diagnosed with Norse in February of 2022, spent 3 weeks in ICU, 3 in a neurology unit, and had 2 weeks of inpatient acute rehab before her discharge home. And, she's here with us today, and hopefully for many years to come, because of the quality of care that she received from one of the best hospital systems here in New York City. She was first seen at a level 1 trauma center before she was transferred to a neurocritical ICU, and I believe that care ultimately saved her life. But we've encountered many challenges in her recovery from the underlying seronegative autoimmune encephalitis, one of which happened, just when she was transferred to her acute rehab facility. That was the first time she was treated intensively without a pure neurology focus. And it's my impression that the team there, as dedicated as they were and as helpful as they were, didn't fully understand her brain injury or the cognitive difficulties she was facing. So I don't think that they were appropriately aware of the needs of Norse patients specifically. And that was a cycle that repeated on her discharge home. We didn't have enough guidance, I think, about how to manage her long-term care. We were connected to in-home nursing care through a hospital social worker, but we didn't have as much guidance or advice as we needed about managing Norse and all of its sort of after echoes in our everyday life. She received in-home visits from non-specialist OTs, PTs, SLPs, as we built up a network of neurologists to sort of help her through all of the sequelae from the original injury. She suffered for many months from this idiopathic refractory nausea that was so debilitating that she would spend hours curled up on the couch, and eventually led to her considering suicide. And I think it's important to say on this call that she was ready to kill herself to get rid of this nausea. And that was something that we weren't expecting at all. She would have breakthrough seizures despite her medications. The nausea eventually dissipated as mysteriously as it arrived, but she worked through several other autonomic challenges like temperature dysregulation and acne, paranoid thinking, depression, perseveration, and random things like muscle pulls from her long-term hospitalization, and then side effects from medication that required rehospitalization. One medicine ended up giving her methylhemoglobinemia, which is a weird one. She had severe constipation for several months, which is beginning to abate. And none of these things were really described to us or discussed with us as potential outcomes. About a year after her onset, she returned for an EMU study that confirmed a diagnosis of epilepsy. And though she hasn't had a seizure in over a year, many of the other impacts of that epilepsy touch our lives from day to day. You know, she gets stuck in negative thought patterns. She's emotionally absent, lacks awareness of other people, has difficulty completing tasks, and even initiating tasks. She has apathy and has a tendency to interrupt people. And these things are all very difficult for us day to day. And the point I think I'm trying to make is that though she survived, I've lost the wife I had, and she lost a sense of herself. Negotiating that grief has taken considerable energy on our part. We're both in individual therapy, and we're working with a couples counselor, things that we do outside of her pure neurology follow-ups. You know, before this happened, she was a VP level communications expert at a major hospitality company. She spent a year on medical leave, but returned to a part-time role working about 15 hours a week, and it frankly didn't go well. Between her limitations and the needs of the workplace, she ended up losing that job. Her doctors are sort of recommending against full-time work, given her difficulties with understanding and processing new information. They have concerns about her abilities to manage stress, and will she have the stamina for part-time employment? And I think it's a difficult question or conversation to have with the patient. How do you tell them that their disability means that their work is not going to be the same as it was? You know, my wife and I live in this state of suspended animation, grieving for the past, surviving the present, and hesitantly planning for an uncertain future. We found a lot of support through programs such as Hopscotch out of Dartmouth, which has helped her learn compensatory strategies that increase her independence. We ended up finding a great speech pathologist who had specific experience with autoimmune encephalitis patients and was able to approach my wife with a deeper understanding of some of her underlying injuries. Communities have been very helpful to us that we've met through the Norse Institute and also through the Autoimmune Encephalitis Alliance. And these programs and networks, I think, need to be spoken about more with patient families because they've been a true lifeline for me. Obviously, many of the doctors we've seen along the way have rarely dealt with Norse patients or even autoimmune encephalitis patients. You know, one doctor early on told me that he'd seen 3 cases in his entire career. So it's a very lonely road. And the impact of that solitude on the patient and on the family is very real and needs its own treatment and management. So I appreciate the chance to come here and remind you of the impact that Norse can have on the real people who suffer its devastating impacts, even when the outcome is, in our case, a fairly favorable one. I think patients and families need more information sooner about what to expect, even if the long-term outcomes of this are poorly understood. You know, the rehab facilities we dealt with, the post-critical follow-up practitioners, they need a better orientation to how to care for patients with these sorts of injuries. And I think more than anything, we need a better language for talking about Norse in general, whether it's with a physical therapist, an insurer, a disability claims administrator, an employer, or just a friend. The facts are working against us in the fact that Norse is rare and it's poorly understood, but we need a better way to talk about it that can immediately telegraph the urgency and the seriousness of this and its aftermath. We need better storytelling as well as clinical solutions and scientific breakthroughs. And in fact, I was heartened to see Dr. deCampo using the term emergent storyline on one of her slides just now. So I thank you for everything you're doing. And my thanks to Nora for the opportunity to come and share just a little bit about the patient experience. And with that, I say thank you.
Thank you very much, Paul, for sharing all that. A lot of that, certainly everyone in the Norse Institute is now well aware of, as we're trying hard to measure these things as outcome measures in any future studies we're doing as well. Sounds like you and your wife have gone through a lot of the common long-term cognitive mood type of things, but also sounds like you've had some less common issues as well. But yeah, those are fantastic points, and we may come back to you for further advice at some point. All right. We're a little behind, but we're still going to take our 5-minute break because that's the one thing we never want to remove. So we'll come back at 11:06 for Dr. Helbig's presentation. See you all in a few minutes.
All right, Larry, I'm already unmuted.
Okay. Sounds good. All right, it's 11:06. So welcome back, everyone, for those of you who went away. Okay, so our next speaker, you've already heard from him, but now I'll introduce him. It's Ingo Helbig. So he's did his training, a combination of in Germany and in Children's Hospital Philadelphia, in pediatric neurology and epilepsy, and he also did a research fellowship in Melbourne, Australia. He's now a famous neurology attending and geneticist, as well as director of genomic science at CHOP, and he's going to be talking to us about computational phenotyping and data integration in FIRES and Norse. Ingo, it's all yours.
Thank you, Larry. Can you repeat? Can you guys hear me?
So, I'm sorry, say that again.
Just repeat the famous part again because my audio
Oh,
was
yes, yeah.
kidding. So you guys can hear me and you can see my screen?
Yes, I see the overview slide.
So I'm going to talk about, give me 1 second. I'm going to talk about electronic medical records, computational phenotyping, and data integration. And this is actually a very nice follow-up to Danielle's talk because this kind of gives you the other side of genomic data. And this is kind of the clinical data that comes along with it. So I'm going to give you an introduction into phenotypes in the epilepsies and how we're basically running into problems when we try to understand clinical features at scale, not in an individual patient or in a small patient series, but if you want to look at a larger patient cohort. I'll talk about this mysterious term data harmonization. And then I want to give you some insight onto what is happening on the in the data standardization field when we use standardized entry forms and standardized analyses. And then finally, I'm going to dive deep into the medical records and kind of show you what we can do when we try to kind of mine and find as much data on FIRES Norse as we can. So let me get started and talk about phenotypes in the epilepsies. So I'm basically continuing the story that Danielle already started with, this kind of explosive gene discovery in the epilepsies. You can see here that this went in different phases, and especially since we've done exome and genome sequencing, there have been a discovery of maybe 2 or 3 bona fide epilepsy genes per month. However, this kind of growth in gene discovery has left behind the phenotyping field. And when I became faculty at Penn, Dick Lindhout, who's kind of one of the very senior scientists in the Netherlands working on epilepsy genetics, and I put together this editorial that kind of was entitled Advancing the Phenome Alongside Genome Studies in the Epilepsies. And this was basically a commentary on a paper that one of the large epilepsy genetics consortia put out, where they actually found some very unusual phenotypic findings when they just looked at scale. This is what my lab has been doing. So I've been involved in epilepsy gene discovery for the last 15 years, and even 20 years. I'm getting older, I'm realizing. And when we kind of came to CHOP, we had this kind of thought is, do we want to kind of continue what people typically do in the field? And this is going to be a very active and important activity moving on to clinical trials and precision medicine. However, we're sitting on this kind of wealth of phenotypic data and had some ideas. So we actually took a detour and dived into what is called phenotype science. So when we think about phenotypes, probably you guys all have different ideas or different concepts on what this might mean. But I would like to kind of compare this to other features or other types of data that we have in brain sciences or in neurosciences. And what I'm comparing here is a raster plot of neuronal firing. And on the right, you can see AHR counters. X-axis is patient age, Y-axis is each individual, and every horizontal line is a single individual. And while these patterns are not quite similar, you can say we can probably use the same methods to look at longitudinal data in the electronic medical records as we use for analysis of EEG data or other electrophysiological data. So let me just kind of introduce you to this question. How does an EEG compare to phenotype? So we look at medical records. And here's my challenge for you. And this kind of gets to the crux of it. So think about that we increase the number of the electrodes by the factor of 100. In phenotype land, we call this dimensionality of phenotypic data. Then think about that each single electrode drops out for 95% of the time. This is a so-called open world assumption. How do we deal with data that is actually not there because it has not been assessed, or it might be negative? And third, just imagine we kind of remove entire age ranges from the recording. And this is kind of a reference to what's called EMR usage. The fact when we look at phenotypic data, there might be entire episodes where an individual is not present in the medical records because this individual was taken care of at a different center or in a different healthcare system. So putting this all together means that when we compare EEG research or imaging research and EMR research, so phenotype research, we know that 90% of looking at EEG or imaging is basically advanced analysis, having new methods, new tools to really kind of get more information out of this data. However, when it comes to phenotype data, 90% of what we do is data harmonization and making sure everybody speaks the same language. Again, here, where this is the kind of this explosive growth of genetic etiologies in the epilepsies. And again, this phenotypic bottleneck I was talking about. Genotypes are actually quite easy. And this is not meant to kind of diminish any of the work that people in genomics are doing, but they actually operate within very clear parameters. Genotypes can be generated at scales. We have now more than 60 Norse fires genomes. If we had 6,000 biosamples, sequencing them and analyzing them is not a conceptually new thing. It is just a matter of scale and effort. Genetic data is highly standardized. So you could see this from Danielle talk that actually we have very clear coordinates for every single genetic change in the human genome. We know exactly where it is and how we can basically name this. Genetic data is 1 or 2 dimensional. You have 1 chromosome and 1 variant. And finally, the only reason why we could actually analyze the genome data is because we didn't actually analyze genomes. We analyzed the digital representation of a genome. This is one thing that we often don't think about, is that when we think about exome data or genome data, we're not talking about the DNA in this person's body. We're talking about a digital file that we can transfer, that can be kind of transferred from Yale to Philadelphia and can be analyzed here. So all these features is quite a bit in contrast to what we see in phenotypes. They often require manual curation. How we explain things is not standardized. This data is sparse and highly dimensional. That means we have lots of kind of categories, but only few individuals who have a single category. And traditionally, phenotype data is non-computable. So it's basically a very, very difficult situation with many different concepts, many different languages, and it's basically a big Tower of Babel that we have to unpack. So what I would like you to kind of show you now is how we've approached this problem over the last 5 years and how we've kind of built up to the analysis that we finally did in Norse and FIRES. And I would like to use this time to really kind of go into the basis on how we think about categories of clinical data and how we can use all this information we have in the epilepsies to really maximize what we need. So I'm going to introduce the concept of the human phenotype ontology. So many of you who have done biomedical research know gene ontologies, or maybe even kind of other types of ontologies in the clinical space. And what an ontology is basically a dictionary with defined relationships. For example, what you can see here is that is a big tree, and we have different clinical features here. And we know, for example, that an early onset absence seizure is an absence seizure, and an absence seizure is a generalized onset seizure. So all the clinical features that we know are somewhere on this predefined phenotypic tree and have defined relationships. And those are typically relationships where certain feature is a subcategory of a different feature. So this by itself doesn't mean much. This is not helpful other than just kind of knowing where phenotypes are. But you can actually already kind of try to optimize this on this level. So this is David here, who's one of my PhD students from the UK. And what David did in 2021 is actually align this dictionary, this ontology, to the most recent ILAE classification. What we showed is that we can actually increase data capture by 30% if we kind of use this new ontology. And here on the left, you can see the concept of how these kind of dictionaries work. We have certain aspects of the medical records where we kind of talked about certain features that patients have. For example, febrile seizures at the age of 18 months, or polyspike waves on EEG. And all these different terms, they refer to common concepts. And in the middle, you can see these so-called HPO terms, the human phenotype ontology terms. And you can now imagine that actually once we have mapped this, so this little arrow shows the mapping of a term or of a concept towards a certain term, we can almost delete the text here and just analyze this kind of code, this HP code. So here's an idea that we can actually use clinical data, clinical information, and find a common dictionary that we use. We use the HPO because it is a very clear and easy dictionary. There are other dictionaries out there. You've heard maybe about SNOMED. This is a very kind of complex network, but they require quite a bit of refining, curation to make them really applicable to our epilepsies. And the ILAE is currently working on this. So once you have these ontologies, they're actually very nice things you can do. And this should just give you an idea that I'll kind of pick up on again later, that we can now take medical records and find a common language, how patients develop over time. Here's an example now of two different individuals. On the left, we have an individual at the age of 6 months who has infantile spasms and developmental regression. On the right, we have an individual who is at 7.5 months, has febrile seizures and gross motor development. So here we can say, we sense that there's something in common. They both have seizures, they both have developmental features, but how could a computer, how could an algorithm that analyzes tens of thousands of data sets possibly realize that there's something in common here? And this is what we're showing here. You can basically map this now to the HPO terms. You can kind of, or it's called propagate the HPO terms, basically add all the terms that are higher in this tree, and then here bin them into kind of certain time bins. And you can see here that these clinical features are now transformed into these little blocks, and that these patients share some of these blocks, but not all of them. They share the blocks that they have seizures and that they have neurodevelopmental features, but they don't share the fact that they have spasms or febrile seizures. So this allows us now to compare this. So the clinical dictionary, the phenotype ontology, is a very, very complex tool and currently contains more than 16,000 different clinical features. So all clinical features that a human may have are contained in this. And this has been shown here in this figure. So those are all phenotypic abnormalities related to muscular or neurological disorders. And the seizure terms are up here within all the relationships. And you can kind of realize the new ILAE language here already. And this shows you the relationship between these terms. So when we have algorithms that kind of look at relationships between terms, they basically jump through these kind of lines here and make inferences. So let me kind of show you how this can be used in a hypothetical example. So here, for example, we have a number of 100 individuals who have certain clinical features. So you can have focal impaired awareness seizures, maybe 12 individuals, seizures in 82 individuals, and CNS phenotypes, and basically all of them. And you can see it like the number and the number or the degree of blueness kind of shows you how specific a certain term is. Specific means how unique this term is in a certain cohort. You can now take these terms and use this kind of ontology tree in the background and put this up there. And now all these terms are connected. You can see the focal impaired awareness seizures are here. Those are focal seizures, which are seizures and which are CNS phenotypes. And what IC means is the information content. So we typically think about how valuable information is, and information content is actually an engineering term. It's just from information theory, which means it's basically the negative log 2 of the frequency. It means the higher the information content, the more valuable this information is. You can now use this kind of structure. We've now mapped the entire cohort of 100 people on this tree, and this now helps us to go through this ontological tree and look at the relationships. Here's, for example, the question. We have one individual in red who has focal impaired awareness seizures. And then we have individual 2 who has absence seizures. How are these two individuals related? Well, they are related because they have seizures. And in this tree, this is being kind of inferred to the fact that this is the most informative common ancestor, or MICA. And you can now see how you can use this concept to realize that these two people are more related than an individual with absence seizures and an individual with developmental delay. Because if I were to walk up here, I would have the CNS phenotypes as the kind of most informative common ancestor. So if you have clinical data mapped onto these dictionaries, you can compare, you can harmonize, and you have a common language to understand phenotypes. So let me move on and think about standardized clinical data. So I've given you now a flavor on how we can handle data that is already standardized, but how does this data get there in the first place? What we've been doing for the last, now probably 5 years, is to collect clinical data in our kind of epilepsy clinics through a standardized form. This is called a smart form, or a kind of a standardized data entry sheet. And what this is basically a kind of template where you can click buttons, and these buttons automatically deposit this information into the medical record, not as a free text, but as a very defined element. So for example, seizure frequency more than 6 months has a very unique address in the medical record or in the medical record database, and we can pull this out, and we know this individual on a certain day had more than 1 seizure per 6 months. We were very lucky that this we could implement this throughout our entire division, and this is data that's going up until December 2022, so we haven't even captured the last year, and we know that we have almost 9,000 individual visits doing routine patient care where we have collected data in a standardized form. So more than 8,000 visits where we have defined seizure frequency data on our patients. So this now opens up a completely new way on how we can look at clinical data. And what I'm going to show you here, and this is now based on roughly 500 patient encounters, is seizure frequencies in a pediatric epilepsy cohort. And we're going to build up to FIRES and Norse slowly, but I wanted to show you how this data looks like in real life. So here is now a comparison of patients with seizure frequencies at their first encounter and then at their second encounter. And you can see these different categories here: seizures that are more frequent than once per day, once per week, and in the end, patients have been seizure-free for more than 2 years. Now we compare the first encounter for every individual with the second encounter, and these lines give you 3 types of information. If the seizure frequency stays the same, the connecting bar is gray. If seizures get better, it's in green. If seizures get worse, it's in red. Now, what you can see here, for many individuals, we see the same seizure frequency, but there are quite a few changes. So this is called an alluvial plot and has become our standard way of analyzing this clinical data that we get from the medical records and get an idea on how seizure frequencies happen over time. So let me just leave this up for 1 second. This is our general framework now, our general template to handle phenotype data. Now let's move in a bit more detail. Now let's look at the different types of epilepsy that we deal with. Here we compare Lennox-Gastaut syndrome, Dravet syndrome, and childhood absence epilepsy. And you can immediately see that there are different patterns. We can see, for example, patients with Lennox-Gastaut syndrome, they have multiple daily seizures in their first and their second encounters. Patients with Dravet syndrome have many green and red collecting lines, whereas patients with childhood epilepsy tend to get better. You can see the dark red bar is much, much smaller in the second encounter, and there are lots of green connectors. So this is a new way to look at seizure frequencies, and this gives you an idea on how we can basically now reconstruct a patient's journey through the entire care history. So now I'm going to jump quite a few steps forward. Now I'm going to actually jump to some examples in other genetic epilepsies where we've done this really at scale. Here's our publication from in Brain from December 2023. Again, here you can see this alluvial plot. This is now data that looks at 162 individuals with a rare genetic epilepsy, STXBP1, looking at seizure frequencies that were reconstructed for every single month across 1,600 patient years. So we pretended that we had seizure frequency data. We reconstructed this from a medical record and pretended we had used the smartphone every single month. Now you can use this as a general concept. And you can see here the seizure frequencies in all the different individuals and here how seizures change. This is a different genetic epilepsies called SYNGAP1-related disorders. And you can see again here this kind of pattern of seizure frequency. And this gives you an idea on how SYNGAP1 presents, that there's very few seizures before the age of 1.5, and then the seizures rapidly increase. Here are two examples of medication landscapes. So in the same time, we can go into the medical records, pull out every single medication, prescription, and administration, and see how these medications are changing over time for seizure medications and behavioral medications. We can use the same concept now and look at epilepsy surgery. Here, this is a data from roughly 90 individuals who had epilepsy surgery. You can see all the different seizure frequencies going up, building up slowly to the time of surgery, and then many patients remain seizure-free. So those are different examples on a real-time epilepsy clinic, on 2 genetic syndromes, and here on epilepsy surgery. But this gives you an idea how we can, analyze clinical data in a new way. Here's now basically kind of the latest iteration from this. This is now STXBP1. You've seen this plot here with the seizure frequency. We can now combine this with developmental data that we get from medical records and from medical record aggregators, for example, Citizen. And here we can combine this with standardized scales that we kind of get from our analysis. We can even now move further and say, if we have this data, if we have more than 1,500 patient years where we have seizure frequencies that were kind of, analyzed on every single month, we can go back and forth and simulate clinical trials. We can ask the question, if we now hypothesize that there's a medication that would reduce seizures by 20%, what are the windows when we can see this? And what we've seen with STXBP1, which is a very mysterious condition, we know that there are 2 windows. We will have a very high success of doing a clinical trial because we can see a very small difference. So this was my kind of brief introduction of the new language for phenotypes. We think about ontologies where we have standardized data on a large phenotypic tree. Then we have ways on how we can analyze severity and analysis over time. Now let's move on to the medical records. So I've prepared you now to kind of for some of the tools that we need. Now let's put this into action. Larry, I don't know if I've shown this to you. Do you know what this is?
No.
So this is the following. X-axis is patient age. This is 0. This is 10. Y-axis is patients that are stacked by their first encounter in the medical records. Every horizontal line is a single patient. Every single dot is a single patient encounter. What you're looking at here is my entire division. Now we're looking at more than 1 million patient nodes, more than 100,000 individuals, and more than 123,000 patient years combined. So this is the entire child neurology clinic at Children's Hospital of Philadelphia. You can see there's a little kind of kink here. This is the age of 18. We're a pediatric institution. There are still some individuals who we kind of see, but many of our patients are young. And by the way, this plot is now almost 7 years old. So this is a rapidly aggregating data set. And what I'm using this for is an example that data in rare conditions, data in FIRES and Norse, is not rare. Our understanding of this is rare. This data is there. We just need to learn how to read it. One thing that we have done is taking all these medical records and systematically translating them into the HPO terms I've just showed you. We've taken natural language processing, taken 53 million patient notes, and just on 1.5 million individuals, and kind of transformed them systematically note by note by note into human phenotype ontology terms. Now we have standardized terms for every single patient encounter on a large number of patients. And this gives you an example now of what we can do. We look at status epilepticus, and this is now in a Dravet syndrome, which is a genetic epilepsy that is relatively common, but it's not the most common condition. But here what we can see is we can reconstruct phenotypic data of more than 3,500 patient years and look at the frequency of status epilepticus on a month-by-month basis in patients with Dravet and patients without Dravet. This is already quite close to where we want to go with FIRES and Norse because we look at status. But this gives you the kind of granularity and the kind of power of what we can do with medical record data. Here's an example on how this natural language processing works. I'm going to be using a hypothetical example of a patient note, and this patient note is being parsed by our natural language processing algorithms and is being mapped to specific HPO terms. And again, once we know what these terms are, and once we know what the kind of age of the patient is, we can take all these other things out and just use these terms to do our analysis. This analysis, what we can do with this is extremely powerful. For example, here, this is currently a paper that is under review. We can actually show that in Dravet syndrome, if we delete any patient note that has been made after the diagnosis, really look at this kind of small patient history before a diagnosis is made, we actually can anticipate a genetic diagnosis of Dravet syndrome by almost 6 months. This is already very clear with a more than almost 90% accuracy that we find these patients through standardized medical record data. So here's our toolkit, natural language processing. We can dive into the medical records and can go in there. Now let's see what we do with FIRES and Norse. So Danielle has already kind of showed this slide, but I want to kind of now make this a bit more granular. So we have 53 million patient notes. We went through every patient note and asked the question, does this patient note contain the term febrile infection-related epilepsy syndrome, FIRES with seizures or status epilepticus, or Norse with febrile infection? So we went through every single patient note, and we have a database that allows us to do this very, very rapidly. And first of all, we find lots of artifacts. So we have 201 individuals, and this is basically now at this time point where we can say this is a number of notes or number of patient charts that we can go through manually. We initially kind of filtered out some artifacts, and medical records in the same way as genome data is lots of uncertain variation, lots of artifacts. And now we had 59 individuals that we went through manually. And then we basically find 25 individuals who had a confirmed diagnosis of FIRES based on the formal diagnostic guidelines. So this is now actually, this is the best way, in my opinion, how you can use medical record data, the combination of data analysis and then manual chart review. This is really the kind of the sweet spot in having large data speak for you, but also having the expertise to review these charts. And this was a kind of major lift. But this basically now gives us the entirety of all patients with FIRES and Norse kind of seen in our system. So what can we do with this now? So we can give you some general ideas on how these patients were actually kind of treated and when genetic testing was done. And Danielle already referred to this and how our patient flow was. And I must say we've only kind of started analyzing this data. We're kind of far from finished with this, but this gives you a rough idea on how heterogeneous our patients are when it comes to FIRES and Norse. Here on our next slide is just an idea of what the treatments were. So we kind of really looked at our patients from every year, starting in 2010 to 2021, when we kind of finished our study, and looked at the kind of specific treatments. So basically everybody got almost steroids, lots of IVIG, but you can see that the use of Plex and ketogenic diet only really happened starting in 2016. Yeah, so there are some kind of tendencies on how we kind of treat Norse and fires, and you can see, especially in the last few years, patients have gotten like all different therapies. Here's again the kind of Danielle slide on the amount of genetic testing is done. So in pediatric institutions, especially genetic testing is very close to care. There are some pathways how we can do inpatient testing. We're involved in some of the kind of strategies on an institutional level to really kind of bring this to patient care. We have now standardized newborn sequencing on a genome level, and we have very clear protocols on how we can get testing done in patients who are critically sick. And this has been done for the last 10 years. There are very few patients with fires, especially at larger institutions, who do not have genetic testing. We know that the yield of genetic testing is low, but what I typically tell my colleagues is there's not really any indication where we would ever kind of rely on this. We would like to exclude metabolic conditions. We would like to make sure that a patient does not have POLG, so we can use deCampo. Yeah? But again, testing is being done on a regular scale. So while the genetic yield for fires in Norse is relatively low, and we have these few genes that we're really kind of now focused on, let's take a step back and look at a larger cohort of patients with status epilepticus and refractory status epilepticus. And this is an important component. Again, we kind of used our natural language processing and kind of went into the medical records and looked at all patients with status epilepticus. So the most common gene in any individual with in any cohort with status epilepticus is SCN1A. Dravet syndrome typically presents with febrile status epilepticus in the first year of life. And here you can see the common epilepsy genes, SCN1A, KCNQ2, SCN2A, 8A, and DEPDEC5. Yeah, and you can see here the number of individuals we have in our study. And now let's contrast this with patients who do not only have status epilepticus, but refractory status epilepticus. And this is where the genes come up again that Danielle mentioned. So RANBP2, one of the most mysterious genes that I know, CACNA1A, a gene that we've kind of really dealt with repeatedly over the last 5 years, and we're currently the kind of center at the East Coast to see most patients. PCDH19, which is a very prominent gene that often presents with almost an encephalitis-like presentation, and KCNA2, which also can cause a very severe status epilepticus. So this is now the snapshot of status epilepticus and refractory status epilepticus, parsing for genetic diagnosis using this kind of large data set of more than 30,000 patients with epilepsy that we have in our database. So now let's take this one step further. Let's ask the question, when we're kind of trying to circle in on Norse and fires, can we see some patterns in patients who have genetic diagnoses and patients who don't have genetic diagnoses? And what I want to explain to you right now is basically this plot here, which is called a phenogram. This is one of these analytical visualizations we developed a few years ago. On the x-axis, you see the frequency of phenotypes. Those are all categories that we see in clinical features. Those are more than 2,000 different categories. Every dot is a single phenotypic category, single HPO term. Those are the frequencies in individuals with status epilepticus without a genetic diagnosis. Those are roughly 750 individuals. On the y-axis, you can see the frequency of the same terms in patients with a genetic diagnosis and status epilepticus. Yeah, and here you can see these features that stand out. So everything that is significant at a 0.05 nominal significant level is in red. And the more a term deviates to the top left, the more it is associated with a genetic diagnosis and status epilepticus. And we've labeled a few terms here: intellectual disability, hypotonia, infantile spasms, developmental delays. So we know that patients who have status epilepticus and are found to have a genetic diagnosis have a much higher chance of having a comorbidity of other neurological features. Yeah. For example, on the other hand, status epilepticus without, we see that patients are typically mild, that they have ADHD, they may have unusual infections, and they may have more pain-related issues with this. So here's a nice way how we can use this large data. Those are several 100,000 patient years and basically millions of data points that went into this. And we're comparing these kind of two paragraphs here. This gives us a readout on how status epilepticus presents on a population level. So this was my little whirlwind tour through computational phenotypes and EMR analysis. I gave you some idea on how we can, try to approach the challenges that we have in phenotypes by first developing a standardized language. Then I kind of give you an idea on how we need basically standardized data entry and how we can even mimic standardized data entry to reconstruct larger data sets. And then finally, we applied this all to the medical records to kind of get some in-depth knowledge on our patients with FIRES and Norse. And with this, I also want to kind of thank my team. I stole this slide from Danielle, so I apologize, and I'm happy to take questions.
Thanks, Ingo. It's fantastic. If people have questions, you can raise your hand or put them in the chat. We have a couple of minutes. I'll ask one or two. Do you think, I know sometimes with scribes and things now, visits are just being recorded. It seems like if we stick to just what's in the notes, we lose a lot of information. Should we just be storing the actual recordings of everything that's said in a visit?
So one of the rules of phenotype analysis, Larry, is you get what you get and you don't get upset. Yeah, so this is one of these things where we have to say, of course, it would be nice if we had standardized data. Just to give you an idea, our kind of standardized epilepsy forms, even though this is really kind of mandated on an institution level right now, is only really kind of used in maybe like 50% of all epilepsy visits. Yeah, so what we have, there are always limitations with lack of data. At this point, I would probably try to rather raise the bar for the minimum of data entry. Yeah, I would rather have patient or physicians have standardized reporting of seizure frequencies rather to go more in depth for individual visits.
I'm just imagining something that doesn't rely on the doctor doing anything properly, just basically recording the whole thing.
Yeah, it's so the idea. There's lots of implementation science behind this, how you can actually make this easy. I think we're at a sweet spot right now where we really kind of have made this easy. We had some kind of early adopters who were kind of quite influential. Yeah, this is really an institution decision where you would have to basically use this. If there's a way to kind of transcribe entire patient visits without actually being forced to rely on the medical records, this would be interesting. There's also lots of artifact in there, and I think once you kind of have recording and use natural speech, I think you have another layer of complexity, and there may be some very clear advantages, but there may also be challenges in there. You filling me from 2 seconds now. Sorry.
Looks like, okay, we have, I see at least 3 questions. So from the chat, one, how widespread is the use of your process across medical centers? And what do you make of the yield of suspicious genes, given the phenotype of Norse patients having no significant medical history?
So there are, so I should mention 2 things, that there are different epilepsy learning health systems. So the Epilepsy Foundation has created the Epilepsy Learning Health System, and some of the standardized data entry is kind of aligned with this. So there are some very large data capture efforts for people with epilepsy to have the standardized data analyzed and deposited. This is often now done for QI purposes. The other effort is in the pediatric field is the Pediatric Epilepsy Learning Health System through kind of Zach Greenspan at Cornell, that kind of specifically looks at analyses in childhood epilepsy. So these tools are being used and are kind of increasingly adopted. And then the second question was yield of suspicious genes given the phenotype of Norse patients. So the yield overall is low. I might want to go back and actually say when we started out doing epilepsy genetics 20 years ago, the yield in any type of epilepsy was low. So when I'm telling you these days that we have a diagnostic yield of 30% in severe childhood epilepsies, this was inconceivable 10, 15 years ago. The fact that we don't have a clear answer for most individuals with kind of Norse or FIRES right now is not what we expected. I remember writing grants 10 years ago saying, will kind of probably have the same diagnostic yield, but it is actually very intriguing. And we're really trying to figure out, what does this tell us about this unique patient population? With the idea is, are there mechanisms at play that we can understand through genetics that we wouldn't necessarily see? So the yield is low. We know that most patients with Norse don't have a prior medical history, neither have patients with kind of fires in the pediatric sphere. But even if only a small percentage of individuals have a genetic diagnosis, this may really help us understand the underlying mechanisms that are very elusive now.
Great. All right. Thank you very much, Ingo. We're we're only 1 minute behind now, so we're going to move on to our next speaker, who is Laura Mantoan Ritter. So Laura,
Hi, Larry.
hi, thanks for joining. I'll give you a 30-second introduction. So Laura's a consultant and adult epilepsy genetics lead at King's College Hospital in London, as well as an adjunct reader at King's College. And her group investigates novel diagnostic and therapeutic tools and precision treatments for super refractory and genetic forms of epilepsy and of Norse. And she's going to talk to us about a proposal of a trial related to vagus nerve stimulation. So Laura, take it away.
Thank you very much. Thank you very much, Larry, for the introduction, and to you and Nora for kindly allowing us this time to talk to you about our study. So I'm not here on my own. Let me just see. Can you see the screen? And you can hear me all right?
Yes,
Great,
perfect.
great, brilliant. So our study is called the investigation of the use of VNS in treating Norse. And this is one of the pictures from the helipad of King's College Hospital, where we admit Norse from the region and across the UK. So our study is funded by the Charles Sykes Memorial Grant from the King's College Hospital charity. I'm the lead PI, but I have many co-investigators, and today with me I have Richard Selway here. I don't know, Richard, whether you want to show your screen. He's our lead epilepsy neurosurgeon here at King's College Hospital. Thank you for joining us today. And then we have James Galloway, who's a consultant rheumatologist, and Matthew Walker, who I'm sure many of you will know at University College London. So as a summary, it will be a multi-center N-of-1 trial series to study the efficacy and mechanism of action of VNS in the treatment of Norse. And because I know there might be some lay members, I've also put a little summary in blue where I make it more a little bit more explanatory. So what we really want to find out if VNS can be used as an additional management in really reducing the severity of seizures, or perhaps even to stop status epilepticus in patients with Norse and virus. So I don't know how many of you are familiar with vagal nerve stimulation. So this is a little advert image from the Sentiva model. As you know, the vagal nerve stimulation consists of a little battery pack that is implanted here in the chest region under the skin, a bit like a pacemaker, and then the leads are tunneled up and wrapped around the left vagus nerve. And the battery pack, the software essentially sends off a constant stimulation, but it can be triggered also by a magnet. And at King's College Hospital, we implant. We are one of the regional centers in the UK for vagal nerve stimulation. So in Norse itself, the background, it's really unknown how it works. It might be directly through modulation of brain activity, through electrical stimulation, or indeed there might be an effect of modulation of the immune system. And Richard and I summarized the available literature in this paper here in Frontiers of Neurology last year. There are about 15 patients published in the literature that have been treated with vagal nerve stimulation, both in the acute and the chronic phase, with varying results, but generally good tolerability. So the second aim really is to establish not only if VNS is effective and can be used as an adjunct, but also if it has an immunomodulatory effect. And indeed, if you could use inflammatory molecules as a biomarker of VNS effect in Norse patients. And just a brief word of why we chose vagal nerve stimulation versus other forms of brain stimulation, such as, the brain stimulation. The main reason is that it is licensed in the United Kingdom. It is NICE approved and therefore funded, and is widespread across many of our tertiary neurosciences centers. And part of it was probably also because its relative ease of implantation, and Richard can tell you about that. And it doesnent have the need to open up intracranially. So these are our scientific hypotheses. We're going to ask if VNS interrupts refractory status by acutely desynchronizing ictal rhythms. In those who don't respond immediately, whether it does have a chronic neuromodulatory effect and reduces the amount of anesthetic used to achieve seizure suppression. And finally, if it influences the inflammatory response, and we're going to measure a variety of inflammatory molecules. Obviously, Norse is a very rare entity in virus, and we are embedding it into Norse UK, which is very much like the Norse Institute in the United States. It's a consortium of all tertiary neurosciences centers who treat Norse, and it was founded by myself and Lena Nashev. And these are some of the aims and objectives we've already achieved, and it's going to be embedded and collaborate with the Norse Institute, which is absolutely exciting. So we are hoping to recruit from these 30 UK centers. Each center sees about 1 or 2 patients a year. So the trial design is prospective, single participants. It's called an N of 1 trial series. The inclusion criteria are adults and children diagnosed with Norse virus according to the known criteria. And we ideally would want to recruit 10 to 30 cases over 5 years. I would like to add that VNS suitability will be discussed very early, so that we have an alert system when a Norse case comes in, and we will discuss VNS implantation early, so 7 to 10 days post-onset, and it will be agreed upon at a joint epilepsy surgery meeting held online. And it will not change in any way the standard management, anticonvulsant, immunosuppressive, and anesthetic management that each center has, and we share the same protocol now across the UK for the entire duration of the study. So this is how an N of 1 trial normally looks like. You typically have a run-in period to the very far left. Then you have a treatment period, A, of about 15 days we established for vagal nerve stimulation. B, you typically would have a washout period where you stop the treatment for about 5 to 7 days, and then a second treatment period to show that it works again. And at the end, a final evaluation at 1 year. Now, normally when we implant VNS in chronic patients with chronic epilepsy, we up-titrate the stimulation parameters over months, up to 6 months. But in Norse cases, and this is an example of one of the cases we treated with Norse at King's, as you can see, it is switched on pretty much straight away in the literature within the first 14 days, and then rapidly up-titrated to a target that you would normally achieve at around 6 months. So within 7 to 14 days. And in the literature, the effect normally is visible within the first 2 weeks of VNS treatment in those patients who respond. During the treatment phase and during the entire duration of the trial, we'll do continuous EEG or spot EEGs and take serum and CSF for inflammatory cytokines before and after VNS implantation. Our primary endpoint, of course, is the acute treatment effect. So we will have responders, those where the status epilepticus stops. In those, it would be unethical to stop, to switch off the VNS. And we will continue VNS stimulation for at least 1 year or as long as clinically indicated. The ABA design will be used for non-responders. And because obviously the patients will get anesthetic sedation, we will use the cumulative amount of sedation as an additional co-endpoint, as a surrogate marker of seizure frequency. And then monthly review of seizure severity and obviously EEGs when available. And I think this was the last slide. I'd like to thank the Norse Institute in particular for this fantastic collaboration and all the people who contribute to Norse UK and our grant funders. And finally, I'd like to end with this final slide with our team here up on the helipad. And it would be absolutely fantastic to be able to expand the trial across the United States or other countries in the world. If you would like to collaborate, please contact me on this email address, and I'm very happy to take any questions. And Richard is here as well for any more technical and surgical related questions.
Fantastic. Thank you, Laura. There's a question about what are the cytokines that are being reviewed?
Well, there will be several, amongst them interleukins, TNF-alpha, HMGB1, and serum ghrelin levels, and others. But those are the main ones.
Okay, and is there a certain number of sites you're looking for, or you'll take as many as people
No,
are
we'll take
interested?
really as many as we can. So obviously in the UK, we will recruit from any center implanting VNS, and we will also accept patients for transfer if they cannot implant VNS themselves. But really any center who has Norse patients or virus patients, or both adult and pediatric, and who can implant VNS, absolutely could join us.
A couple other questions popping up. I don't know if you can see the chat.
No,
VNS,
sharing and see if I can. Oh, yeah, I can. The chat. Yeah.
the last two basically there.
Oh, the last two. Brilliant. So there is Nora asking, is VNS used in the acute phase generally in the UK and in the US? And if there is a discrepancy, what are the reasons? Well, it really depends on which center you essentially are admitted to. I think worldwide there were about 8 cases implanted in the late phase and a bit more in the acute phase. It depends really on the experience of the center. So we with the 2 cases we've had, one in the chronic phase and one in the acute phase, we've had a good effect, and we discuss it early. And that's what I always advise when you get a Norse case and they don't respond, is just at least to consider it early, because you're going to end up having a patient with very drug-resistant epilepsy in the best of cases. So consider it early. And it's just, I think, a general mentality that one has to think about it a bit earlier. And then the final question, Marissa asking, many of our patients
I think that one's for Richard. About
Yes,
whether
yes,
tracheostomy causes additional problems with if you can give a quick answer to that.
absolutely.
Yes, it causes additional problems. Yes, they can be got round. So one of the things I sort of push for is often there's a suggestion of VNS and then there's a bit of a delay while other things go on, during which time a tracheostomy gets put in, the patient gets a bit of a chest infection, and it becomes more of a problem. And one of the advantages of going in early is you can avoid many of those things. But yes, and the other thing is a left-sided central line is often also a bit of a issue, but you can work around it. But obviously it's nicer to preempt those things.
All right, fantastic. Well, we will certainly help you recruit and look forward to collaborating with you on this. All right, I'm just gonna make a few closing remarks before everybody goes about the rest of their day or evening. First, I want to thank everyone who joined, particularly looking at the list of people on here. We have a lot of people from Europe. I think I see UK, Italy, France, other places. We have Costa Rica, Israel, and a special thanks to people from Korea who are joining us somewhere around midnight, maybe after. So it's much appreciated. Couple reminders. One is we are able to take samples from anywhere in the world. I mentioned earlier, and there've been a few questions about that. So just email us at that norse@yale.edu email address. We're also not only in the acute Norse or FIRES patients, but also the chronic stages. And we're also looking for control, so refractory status with known etiology and patients with autoimmune encephalitis without status. Those are some of the specific controls we're looking for. Another reminder is please join the family conference, which is this Wednesday. And then we have our next scientific symposium very soon on May 9th, which will be on metagenomics, autoantibodies, and proteomics, mostly from Michael Wilson's lab at UCSF. And then just wanted to again thank people who helped organize this. So Nora Wong, Maggie Gopal, and Anthony Jimenez. Seems everything ran very smoothly in the end. And a big thanks to our speakers, Danielle, Ingo, Paul, Laura, and Richard for joining us. And thanks to everybody. You're welcome to stay on and chat. That's the end of our formal program. This was recorded. We will have it posted, and contact and all of that will be available on the Norse Institute website as well. So thanks, everybody.